All right, everybody. Today we're going to talk about something really interesting and something that I thoroughly enjoyed researching and learning more about. It's the role of horizontal gene transfer as well as integrons in the acquisition of antibiotic resistance. Now, that's a bunch of words, but we're going to break it down real simple. First, we're going to look at horizontal gene transfer and what integrons are and how they cause an evolution in bacteria, right? And that evolution being antibiotic resistance, which is very relevant to all of us. I would guarantee almost all of us have gone to the doctor and gotten antibiotics for something in our lives before, or we will at some point. So to start out with, horizontal gene transfer is something probably have heard about in your courses biology, and you might remember it more or less, and how it relates to resistance. But integrons are a specific case of horizontal gene transfer, and they're very efficient and they're targeted, which makes them so powerful and often in many ways scary as we look at the future and ahead and what challenges we have in front of us. There's a specific case of A. baumannii that really elucidates these issues that scientists have right now, not just scientists, but all people, whether you understand, whether you're a factory worker or whether you're a doctor. I mean, these are really issues because all of us benefit from antibiotics. Interestingly enough, though, drugs being antibiotics are actually causing an SOS response in these bacterium or in these colonies that might be promoting the resistance itself. We're going to go over that a little bit more in more detail. But certainly more research is needed to understand these integrons, how they behave, how they adapt, how they evolve, and also how to prevent this SOS response. Featured to the right, you can see here you have two bacterium. This one's kind of fun looking, right? But right in between them, they have a sex pilus. This is a type of horizontal gene transfer I'm about to go over, but they're exchanging genetic material, which is what it's all about, right? So overall, horizontal gene transfer is the transfer of genetic material, but the key is that it's of cells, from cell to cell of the same generation. So this isn't parent passing genes on to their daughter cells or anything like that. This is the two daughters exchanging stuff, which is kind of hard to wrap your mind around it, right? I mean, I have a mom and a dad. It's kind of hard to think of me pulling some genes out of my chest and giving them to my brother, right? In our heads, it doesn't work that way. But the cool thing is, it's so fascinating. Bacteria do this, and they do it pretty darn often. And it's usually really beneficial. Sometimes it's not so beneficial. It can even be harmful sometimes. But more often than not, or in any ways the cases we view, it's often really beneficial for them because it has an antibiotic resistance. So in the first case, hyperconjugation. Right here, it's just called conjugation, kind of both ways. This is the one we saw on the slide before. You have a cell up here and a cell down here. They form this pilus, right? He has, this is going a little more detail, but he has the F factor, their fertility, and he can pass off. Usually it's a plasmid being passed off this way. Plasmids very often have the antibiotic resistance. So this is a pretty effective thing that it does. And we've studied this for a long time. It's pretty well known. But we're going to dive into something that may be even a little more effective. So that's the first one, hyperconjugation. It's direct cell-to-cell contact sending plasmids, right? Remember back, you have F plasmids, F prime, HFR. All those come into play with conjugation. Now the next type, transduction, is a little bit different. Actually, it's like almost completely different. This is by a phage. And you got these cool spider-looking things that go from cell to cell. I think they're cool. Bacteria kind of hate them because they usually kill them, right? These bacteriophages, what they do, some of them, there's definitely lots of different types and lots of different mechanisms here. This is a huge simplification, of course. And this image, by the way, is from Michigan State, an article their website posted back in 2011. But this phage, it comes in, it injects its DNA. It's got a whole mechanism for this. But this little tidbit, the sequence of DNA, goes in and it can do a couple things. It can immediately start replicating and forming more phages. It can lyse the cell. But transduction occurs when it makes that prophage, right? When that genome, this little tidbit, if this is the, the cell's genome or plasmid or whatever, this goes in, it inserts itself enzymatically, and then it waits until the time's right, the temperature, the environmental conditions, whatever it's looking for, right? And then it replicates, makes a ton of its own little phages, and then it lyses the cell and goes, right? But when it's making its own phages, it repacks it with this little tidbit of DNA so that it can do it again in the next cell, right? And it's a self-propagating sort of system if it goes unchecked. But when it cuts it out, sometimes it makes a mistake. Sometimes it gets more DNA than it needs. Sometimes it gets less, right? And that, in which case that phage may be ineffective, but sometimes it gets more, and that little extra, then if it gets packaged into the phage right here, goes to the next host, and then it'll insert it. And that can be beneficial sometimes. Sometimes it can be redundant. Sometimes it can be even harmful. Sometimes it can just do nothing, right? If this bacteria, or excuse me, if this phage inserts its genome and interrupts a structural gene, say, for example, in the bacterium, it could end up killing the bacterium, and then it's not very good. So overall, transduction, probably not the most efficient because it's not controlled. It's actually an accident. It's a mistake by the phage when it cuts that out. So as far as transferring beneficial genes, probably not the way to look, right? Transformation, on the other hand, this is where we're going to focus. This is where, to me, crazy stuff happens. I love bacteria just because I think they do things that are so off the wall, like don't even think it's possible, right? This picture shows it pretty well. Actually, here, this is on the outside of the cell. This is on the inside. You have these little, and it shows it, little DNA strands, right? And they come in. So bacteria that can directly take this in are called competent. It takes them in and it can use them. It can put them in their plasmids, whatever, right? Actually, research has shown this is most likely enzymatically done. So this is endocytosis via a transmembrane protein of some sort. That wasn't the focus of this literature review, so didn't cover that intensely, but most likely enzymatically covered or enzymatically performed. But and these pieces of DNA floating out here are called mobile genetic elements. So you might be asking, is that really even, is this often? And that's something what we're going to cover. But what constitutes these little pieces of DNA is interesting. So an integron is a specific type, right? You can just have strands of DNA hanging out there. Integron is not so much just a random thing floating around, right? Integron, this is a little definition that I came up with. I call it the genetic machine because I just think they're so powerful at what they do. And it facilitates the transfer and acquisition of genes. So what do I really mean by that? So what I mean by that is this is a pretty simplified yet good picture of an integron, okay? It's got a gene on here. Well, let's just break down primary components. First thing is right here, you see an INTL gene, okay? That INTL codes for an integrase or a resolvase, kind of some different names for it, but effectively, this is a DNA strand, so it has to be read by a ribosome or something like that, right? By itself, it doesn't do a whole lot. It has to, but it codes for proteins that do really powerful things. Right here, this strand is promoterless. What that means is it doesn't do it on its own, right? It needs some help from a promoter. So the promoter can come in here, bind, and read this way. You have cassettes right here, okay? Now these cassettes actually contain a collection of genes. That's why it's called a gene cassette. Often they're for antibiotics. Actually, more often than not, they're for antibiotics, which is really interesting and something that people are still researching is how did these start? Why do they have so much resistance? Why is it mostly just that on there, right? But this integrase is so interesting, and this is what really makes it powerful. This shows an INTL 1. The 1 classifies the class in this case. Sometimes it's just IN1, sometimes it's INTL 1, depending. Kind of the older research has just as IN. Also in this book by Snyder and Champness. So yeah, so that what that can do, what that integrase can do is it can recognize right here the ATTL sequence and also a sequence on the plasmid. So once this gets in, right, and it gets read, Intel gets formed, ribosome makes it and everything, right? Well, that enzyme now can recognize this, and it can recognize a sequence on the plasmid, and it can actually insert whatever this first gene in the cassette is, right? So that's really cool. But what about all these? Well, that's not the only thing it does, though. That's why it's a resolvase as well, right? Because it can switch these around. Now, admittedly, I don't fully understand. I don't know that anyone fully understands any of this, but that's really powerful. And it goes both ways, right? This picture actually shows it taking up that this gene right here, right, and adding it into its cassette. Then it could shuffle it, put it up here. So this is constantly happening. There's lots of this going on. It's picking up this gene, putting it in, switching it around, going to a next bacterium, and it's effectively just shuffling everything. But the bacteria generally get the good genes, right? So these are vehicles for these resistance genes to go bacterium to bacterium and to confer that resistance. And that's why I said it's so powerful, because it's targeted. Now, there's some other things in here I didn't mention, but I mean, for this to be an effective, it's almost like an operon, honestly, like a lac operon or something like that has a function, except for it lacks a couple functions. You do have the inverted repeat sequences at the end. You have the 59 base pair sequence in the middle. You have a GTT, which is actually, which all serve different purposes for binding of the promoter and all sorts of things. But these are the big parts that you're always going to notice, and these are what they look for when they're trying to distinguish whether or not a bacterium, say like a resistance one, has an integron. So you might ask, well, that's all fine and dandy, but if this is 1 in a million, it's probably not that big of a deal. So how prevalent, maybe a better way of saying, are these? Well, lots of research has been done on that. A specific team back in 2015, they found they took they took some bacteria that they found to be highly resistant, and they tested, okay, in these resistant bacteria, are there integrons? And they found yes, very much so. More so in the gram negative, there were 75% of those resistant bacteria had integrons, while only 50% in gram positive. But that's still a lot. There's definitely a correlation there. This is a cool picture. Everyone that took microbiology has done a gram stain. Really fun, kind of cool. These people did a really good job. Turned out better than mine. But over here, you can see the gram positive, gram negative, right? The crystal violet stain. So definitely prevalent. Definitely our resistance genes, and this isn't the only study, there's a lot out there with them, but this is the one that, I dug into the most here, but it's definitely in these resistant bacteria have these integrons. So we wonder a little bit more like, what their role is and dive into that. Interestingly enough, in this specific study, class 2 integrons, remember I mentioned here the Intel 1, right? So class 2 is going to have kind of a different, more evolved in some way Intel gene. So 5% of all strands had that. So that leads you to the question of what, for the research of what's happening when these evolve? Is that making them more effective, right? Are the ones with class 2 genes more effective, or are they really just what's going on, right? Because if we want to be able to combat this with antibiotics, what's going on here, we need to understand it first. So A. baumannii is a specific example. These are hospital superbugs. And research has shown that in these are very resistant. I mean, even to vancomycin, which is a very powerful antibiotic, 94% in the specific study in 2017, 94% carry the Intel gene. Okay. Now there's a distribution of the different classes here. And this is actually a little bit different of distribution than we saw in different studies, right? So apparently in different bacteria, there's different classes, different things going on. So more and more questions there. Someone once told me a good scientist always leaves the laboratory with more questions than he entered with. So that's definitely what's going on here. But the Intel was prominent. 88% had that. 25% had Intel 1. 23 was 28%. These don't add up to 100. So in these genes, some had both. What's going on there? Can't really tell you. But definitely there's a correlation between the resistance genes and the integron presence, right? It's confirmed by Huang in 2015, or actually I guess this one was first, but either way, they had similar results. 73% of isolates contained integron cassettes. So they tested for the cassettes, which remember is the actual resistance genes, to see which ones had that, right? So a lot of the resistance in these Abelmani are coming from the integrons, right? It's not that they're just born with it, but the integrons are transferring it, right? So very interesting, very big problem. Now, both of these studies were done, or actually one was done in the Middle East and one was done in Asia. And so, and those are areas, if you read headlines, it seems like a lot of the superbugs that we're having trouble killing are coming from those areas. So this is real stuff that's going to be affecting us. I just read a news article about someone in Nevada that was traveling to one of those regions, came back, actually died of a bacterial infection. So very interesting, very prevalent in our lives. So antibiotics, SOS response, promise we cover this. 3 different studies done in relatively close periods of time found that common antibiotics induce an SOS response. So what is an SOS response? Well, to paraphrase, because this is a whole different study, or a whole different topic, SOS response is when a bacteria gets DNA damage. It says, oh, help, and it tries to repair itself, go through mutagenesis, to fix that, right? Now, what they're finding with these specific studies found is that the integron activity is increased by the SOS response. So you quickly see how that's a cascade, right? Someone's got an infection, so you give them some antibiotics. Well, that antibiotic is causing a SOS response, which causes integron activity, which, as we discussed, causes resistance to the very thing that you just gave it. So, I mean, everyone knows that you take more antibiotics, the more we give it out, the more resistance is going to happen. But we're causing them. We could be, if more studies confirm this and we understand it better, we could be inducing a mechanism to cause resistance, not just by happen chance, but causing it. So that raises some flags, at least in my mind. And right here I have the question, are the very drugs we use to fight infections indirectly causing resistance? And that's something that really needs to be considered. So in conclusion, we talked about horizontal gene transfer, the good and the efficient and the not so efficient, the integrons, how they're a powerful mechanism, powerful little machines, as I call them, because they carry those gene cassettes with the resistance, and they can directly insert them and shuffle them and essentially make them mobile. Definitely research is still needed in this area, though, because if we're inducing this SOS response, it'd be really beneficial if we knew how to prevent it, right? Because then those same, potentially, right, or hypothetically, these antibiotics that we're giving someone that's causing resistance, well, we could stop it from causing resistance maybe as fast, like slow that down, so that our antibiotics work and are more effective for longer, right? That would be really key. So the evolution as well of integron classes, we skimmed the surface on this. There's a lot going on with that right now. There's a lot not known, because not all classes are present everywhere. So the evolution of it, how they're changing, why they're changing, what they're doing when they change, as well as maybe some structural stuff of the integron, how we can inhibit or break or destroy integrons, are something that's being studied. Even last week or a couple weeks ago, Dr. Thompson shared with us a link of how horizontal gene transfer may have a role in cancer in people. right? And we don't think of bacteria transferring anything to a eukaryotic cell like we are, but it may have been possible, maybe possible that may account for some of the cancer rates and stuff that we see nowadays. So certainly this is relevant to all of us. Bacteria undeniably are changing. Integrons may be a very key mechanism in how they do that, how they do it so effectively. Um, right now, though, we don't know a lot about it. We need to keep researching it, but less is more. If you don't have to take antibiotics, hopefully your doctor doesn't just prescribe them to you to make you happy because you got some sort of pill that's going to make you magically feel better. Overuse of it is probably the biggest problem we have in it, but it's certainly an interesting topic and certainly something that needs to be explored more. So thoroughly enjoyed researching about this. Here are my resources and bibliography. Hope you'll have a great night.